Now consider the next second classification in biomolecules, and the classification is based on reducing property. Based on what? Reducing property. So based on the reducing property, your carbohydrates can be classified into two: reducing sugar and non-reducing sugar. Okay, listen here. If you have a carbohydrate, listen, have a carbohydrate. If that carbohydrate is able to reduce the mild oxidizing agent, if that carbohydrate is able to reduce the mild oxidizing agent, the mild oxidizing agent can oxidize the carbohydrate, which means your, in alternate sense, your carbohydrate can able to reduce the mild oxidizing agents. Like, I think you know the mild oxidizing agents, say tolerance reagent, Fehling solution. The mild oxidizing agents are tolerance reagent, Fehling solution, and Benedict solution, etc. If your carbohydrate is able to oxidize the mild oxidizing agent like tolerance reagent, Fehling solution, Benedict solution, then to corresponding oxidized product, your carbohydrate become oxidized. Corresponding oxidized product, fine. And if your carbohydrate is able to reduce this mild oxidizing agent, then that carbohydrate is said to be reducing sugar. The carbohydrate is known as reducing sugar, else it is said to be non-reducing sugar. Okay. So, what is a reducing sugar? A carbohydrate which can reduce the mild oxidizing agent like Fehling solution, tolerance reagent, etc. Examples: all monosaccharides are reducing sugar, and disaccharides, all monosaccharides and disaccharides, and disaccharide except sucrose. Sucrose is not a reducing sugar. Okay, I'll explain why the sucrose is not a reducing sugar later. Okay, fine. So one more thing is non-reducing sugar. Non-reducing sugar means if your carbohydrate is able to reduce it's not able to reduce. Carbohydrate not reduce the mild oxidizing agent. That is said to be a non-reducing sugar. Okay. And what kind of carbohydrates are examples for this one means polysaccharides. Polysaccharides, and in your disaccharide, sucrose is not a reducing sugar, so it is nothing but it is a non-reducing sugar. If your sucrose is not a reducing sugar, then it belongs to a non-reducing sugar. Okay, then it belongs to a non-reducing sugar. Clear? So listen, the next, and that's all about the classification. So let's discuss the each carbohydrate one by one. Okay. Now your first carbohydrate is glucose. What it is? Glucose. Okay. Glucose is dextrorotatory, so said to be dextrose. Okay. Glucose is said to be dextrose, and glucose is also known as grape sugar or honey sugar. Grape sugar or honey sugar. Glucose is also known as grape sugar or honey sugar. Ready? Fine. Next, your glucose having a formula C6H12O6. Glucose is a aldohexose. And your glucose can exist as two crystalline form. Can exist as two crystalline form like alpha and beta. We will explain what is the difference between alpha glucose and beta glucose. Okay. And specific rotation. This is dextrorotatory. So specific rotation. This is a dextrocarbohydrate. Specific rotation in aqueous solution is an equilibrium value plus 52.5 degree. Okay. And exist in both open structure and cyclic in aqueous solution. So your glucose can exist in both open and cyclic structure in aqueous solution. Okay, fine. So that's all about the points regarding glucose. Fine. And let's check the preparation of glucose. What about the preparation of glucose? Glucose can be prepared by the inversion of sugar cane. So first one, inversion of sucrose by means of hydrolysis of sucrose. You know, sucrose, the formula cetyl H22O11, which are hydrolyzed in presence of the enzyme called invertase. In presence of enzyme called invertase, and it becomes its isomers like C6 H12O6 plus C6 H12O6. C6 H12O6 plus C6 H12O6. Okay, and this is isomer said to be glucose and fructose. These are the isomer called glucose and fructose, equimolar mixture of glucose and fructose. And by means of fractional crystallization, what it is? Fractional crystallization, we can separate the mixture. By means of fractional crystallization, we can separate the glucose from fructose. Okay, so this is the first method of preparation of glucose. Or another method of preparation of glucose is from listen. The second method is an industrial method from starch. From starch. You know the formula of starch we discussed in the last class. C6 H10O5 all of n times, and this is hydrolyzed in presence of NH2O. This is hydrolyzed in presence of NH2O. Fine. If this is hydrolyzed in presence of NH2O, the conditions required for this hydrolysis is 393 Kelvin. 393 Kelvin with dilute sulfuric acid and a pressure range of 2 to 3 atm pressure. 2 to 3 atm pressure. Okay, fine. So this one hydrolyzed to form N molecule of what? C6 H12O6. So N molecule of glucose you obtain. Ready? N molecule of glucose. Fine. Ready? Okay. Or your starch. This is the starch. When your starch on treatment with the enzyme called amylase, you also obtain the glucose. So these are the method of preparation of glucose. Ready? Fine. And I'll explain the physical property of glucose. That physical property varies with the crystalline forms, say alpha and beta. Okay. So we will discuss the property of alpha and beta when we are in the two different crystalline forms. When we are discussing the alpha and beta. Okay. So that will be discussed later. So moving to the next, structure of glucose. So now the next topic is structure of glucose. Okay. So we know that glucose have two different types of structure. The first one is open structure. Glucose have an open structure. And the open structure is actually the open structure is something like this. There are six carbons like this. There we have six carbon like this. Okay, and six carbons are there. One more. The first carbon is an aldehyde carbon, and four others are secondary alcohol, and the last one is a primary alcohol. Okay, fine. So these are the basic structural requirement of your glucose. Oh, so this can be written in this form also, CHO, where all the secondary alcohol can be clubbed into like this, and finally the CH charge. Fine. And this open structure is analyzed by a lot of experimental reactions. Ready? So let's discuss how the open structure is analyzed using the experimental factor. So let me define some experimental observations based on the structure of glucose. Okay, fine. Listen here carefully. I have the glucose, CHO, CHOH, 4 times CH2H. Okay, let me introduce some evidences for the open structure. The first evidence is that in molecular weight analysis. Okay, in molecular weight analysis, found to be the molecular weight found to be 180 gram mole inverse. 180 gram mole inverse. Okay, so what does it mean? It means that your glucose have a formula of C6H12O6. Then only its formula become 180. Okay, so which ensure the formula. Okay, the formula C6H12O6 confirmed. Okay, so each point have something to say about the glucose to you that you have to keep in mind. Okay, so this mass spectroscopic molecular weight analysis indicates that the glucose have 180 gram mole inverse, which means that the molecular formula is C6H12O6. Ready? A carbohydrate with 180 gram mole inverse should have the molecular formula like this. Fine. The next, that is heating with the hydrogen iodide in red phosphorus. That's a powerful oxidizing agent. So when your glucose on heating with the HI red phosphorus nearly for 50 degrees Celsius, what happens means your glucose turns to normal hexane. Which means your complete polyfunction groups are oxidized to alkane. Well, the alkane is a normal hexane, not an isohexane. We already know that there will be 6 carbon. If it is isohexane like this, then we can realize that the glucose have branched pattern. Where all 6 carbons may have branch if it is isohexane. But here we obtain normal hexane. Since there is a normal hexane formed, it is clear that all the 6 carbons are linearly connected. So by means of this reaction, the idea obtained is all 6 carbon are linearly connected. All 6 carbons are linearly connected in the ring. Okay, that's the point by means of this. Okay, now check what about the functional group. For that, they treated the glucose with this is glucose, right? Yeah, they treated the glucose with hydrogen cyanide. I think you're familiar with the reaction of aldehyde with hydrogen cyanide. You know the reaction of aldehyde with hydrogen cyanide. What happens? what happens? This remains same, CHOH 4 times CH2OH, but the function group aldehyde may react to the hydrogen cyanide to form the cyanohydrin. You know how the cyanohydrin formed. This will be formed. And this is said to be glucose cyanohydrin. Glucose cyanohydrin. So, on prolonged heating with on treatment with the hydrogen cyanide, they forms glucose cyanohydrin. Okay. At the very same time, this aldehyde on this carbonyl compound, this aldehyde on or ketone on this is a carbonyl compound, aldehyde or ketone, whatever it may be, this is a carbonyl compound, which on reaction with the hydroxylamine, what happens? Or during the reaction with the hydroxylamine, there forms oxime. Oxime actually. Structure of oxime will be like this. You know how oxime is formed. 2 hydrogen from here and 1 oxygen from here condensed to form the double bond. And these all reactions which are discussed in aldehydes. Okay. And given in your textbook, aldehydes. So on treatment with the hydroxylamine, this is said to be hydroxylamine. On treatment with the hydroxylamine, glucose forms what? Glucose oxide. Okay, so on treatment with the hydrogen cyanide, form cyanohydrin, but on treatment with the hydroxylamine forms glucose oxide. If you're normally the reaction over the formation of hydrogen cyanide and glucose oxide in presence of carbonyls, it is in presence of carbonyls. Okay, so this indicates that your glucose have a carbonyl group. Doesn't mean that is an aldehyde or ketone. If there is a carbonyl group, then any molecule with a carbonyl group on treatment with the hydrogen cyanide forms cyanohydrin, and any molecule with a carbonyl group on treatment with the hydroxylamine form oxime. So these two reactions just indicated that have carbonyl group. Your glucose have carbonyl functional group. Don't know whether it is aldehyde or ketone. So let's check whether it is aldehyde or ketone. Okay, for that your glucose on oxidation with the bromine water. Okay. Normally, aldehydes on oxidation with the mild oxidizing agent like bromine water, it may oxidize to form the acid. Okay. So here what happened means the bromine water on oxidation with glucose on oxidation with the bromine water. What happens? This one get oxidized. Which one? Oxidation takes place in case of the aldehyde carbon. In case of the aldehyde carbon only. Okay. Bromine water is a mild oxidizing agent that is not able to oxidize the ketone or not able to oxidize the alcohol. It can only oxidize the aldehyde to carboxylic acid. So it is clear that we already have an idea there is a carbonyl group. Now it is clear that since there forms a compound like this, the compound is said to be gluconic acid. Mild oxidation product of glucose is said to be gluconic acid. Since the gluconic acid forms, it is clear that presence of carbonyl, right? We already indicate the presence of carbonyl, or we already identified the presence of carbonyl. The presence of carbonyl is a aldehyde, or it's an aldehyde. The presence of carbonyl is an aldehyde. Ready? Fine. Now the next reaction. Check the next reaction. Now we want to find how much OH is there. So this is oxidation with bromine water. Okay, oxidation with bromine water. The next is acetylation with acetic anhydride. I think you are familiar with the reaction of acetylation in alcohols chapter. When alcohols are acetylated, or when alcohols undergoes acetylation, what happens? Acetates are formed. Acetate forms means the OH H is replaced by acetyl group. So this is exactly formed. This is exactly formed in case of what? Glucose, and there forms glucose pentacetate. Glucose pentacetate. So on acetylation, it forms glucose pentacetate. Pentacetate means presence of, the pentacetate indicates the presence of 5-OH group. Okay. Why it formed pentacetate? Because there is 5-OH group. Okay. So there are 6 carbons and 1 carbon is a carbonyl, and the carbonyl is an aldehyde confirmed. Now the other 5 carbons are OH, having OH because pentacetate is formed. Fine. Now check how many secondary and primary alcohol. Okay. So for that, just an oxidation with the HNO3. HNO3 is a somewhat strong oxidizing agent, and it is able to oxidize the aldehyde to acid and primary alcohol to acid, but it can't oxidize what? A secondary alcohol. So on oxidation with HNO3, the top aldehyde oxidized to acid, but the secondary alcohol remains same, and the bottom primary alcohol is also oxidized to acid. So this is not gluconic acid. Gluconic acid where the bottom portion is primary alcohol. So this is actually glucaric acid. What it is? Glucaric acid or saccharic acid. So this is a molecule called glucaric acid or saccharic acid. Okay. So on oxidation with the HNO3, only primary alcohol changed. Okay. No other alcohol get oxidized, which means only the structural changes. Glucose turns to glucaric acid. They all know that the glucaric acid having COH on the 2 terminal end. Ready? All others are CHOH. So it is perfectly clear that out of 5 OH, 4 OH are secondary. That's why 4 OH are secondary. That's why it not undergoes the oxidation. Ready? Fine. So these are the reactions it may ask, and these all are given in the textbook. It may ask, and you should know that. So you have to study this in this pattern, this reaction in this pattern. What is the change in the structure of glucose when the reactions are taking place? Okay, glucose oxide, glucose pentacetate, glucose cyanohydrine, glucaric acid, gluconic acid, like that. And what information is given by each reaction? Okay, 6 carbons are the normal, 6 carbons are linearly connected. There is a carbonyl group, there is an aldehyde group, there is 5 OH group. Out of 5 OH, one is primary and the other 4 are secondary. So there are some kind of informations based on this reaction that also you have to study. Okay. So this is the open structure of glucose, and these all reactions are supporting the open structure. Ready?